Good morning or good afternoon, whenever you're listening to this, guys. I hope everybody is doing okay. Today I'm going to lecture on enzyme kinetics. My name is Devin Boland. I am a TA for the Tuesday and Thursday lab sections. So Monday, Wednesday, if you've never had me before, bear with me through this. I hope everybody is doing okay. And let's talk about some McKellies-Menten enzyme kinetics. So why do we care about enzymes? Well, we care about them mostly, and by first defining them as biological catalysts. And these make reactions of life possible, right? And there's many different facets in which enzymes affect both you and I. And first and foremost, for a lot of you who are pre-med or who at least care about medicinal purposes, is disease, right? So enzymes can cause diseases. They can be their upregulation or downregulation can be the result of a disease, and many medicines and drugs are designed to inhibit, affect, bind, repress transcription, and things of that nature of enzymes, right? For an industrial standpoint, right, when you think of chemical engineering, for instance, classical chemical engineering that you would have been taught in like organic chemistry, you would have heard of these huge reactors where they would be at high temperatures, high pressures. Their catalysts are non-biological, so they are things like heavy transition metals, right? These are very pricey and are very dangerous to maintain at high temperature and pressure, right? Take a lot of money, a lot of training, a lot of time, right? However, if you use an enzyme as a catalyst, right, you can think of it as kind of like a bioreactor, right? These react on scales of temperature that are much lower, much more close to physiological temperatures and pressure. And the best thing about this enzyme is that it's regenerated, unlike things like a palladium, your palladium converter in your car, right? That wears over time and you have to replace it, and it's very expensive, right? Another process is things like the environment, right? For remediation, biosensors, and unfortunately, military processes, right? This is a huge thing. Enzymes play a huge role in bioterrorism, right? Any agents or diseases or viruses or infections, things of that nature, can be released or designed to inhibit particular enzymes or attack particular enzymes that can be detrimental to your health. These can also be things such as like chemical warfare agents, right, for both offense and defense. But one of the bigger things here, right, at A&M is agriculture, right? So food processing and meat production, right? Targeting key enzymes can help increase both food processing times and rates, along with meat production as well, right? You can think in terms of like different genetically modified organisms, like cows have been selectively bred over time to put on large amounts of muscle and fat, more so than typical cows. That way you maximize the amount of meat you get per, animal. Other things like genetically modified organisms, some of that cow example, things like crops, corn, wheat, even, St. Augustine grass is genetically modified organisms designed right here in Texas A&M. And then also things like alcohol, yeast, and bread, right? And for instance, right, enzymes can be targeted and modified in crops to impart herbicide resistance, right? That's certainly the case for glyphosate-resistant crops from Monsanto Corporation, right? So enzymes affect almost every aspect of our lives. So the goal of your research paper, right? What is the effect of the single amino acid mutation from changing a single nucleotide, right? That's the whole point of this whole lab, right? Before all of this happened, right? You were in lab, you were doing mutagenesis, DNA plasmid purification, you were doing restriction digests, you then moved on to expression, right? Recombinant gene expression. You moved on to and did purification, right? Before spring break. And that was just to isolate both the wild type and that single amino acid mutation of ribonuclease assay, right? So we want to determine whether altering the charge of RNase assay changes the activity and or stability of that enzyme, right? So why could changing the amino acid alter the activity of an enzyme, right? Well, it could affect many things. First and foremost would be folding and stability, right? Thinking about it purely from the way it folds and what it looks like, right? You could change the entire shape of the protein, although unlikely. It could, your change could be an important region such as an active site, which may be the same, but may change accessibility. And what do we mean by that? That would be accessibility by the enzyme's substrate, right? Or its ligand, the thing it binds to, right? If you change a small residue, you can completely change the local chemical environment of that active site, and now your substrate can no longer bind. This is for the case in glyphosate-resistant crops. There was a single amino acid change, and it prevented the endogenous substrate for the particular enzyme in plants that bind a precursor for amino acid biosynthesis And instead, glyphosate being bigger was prevented from binding. So we have glyphosate-resistant crops, right? Or protein may be more stable and can be too stable, right? As is the case kind of for RNase SA wild type. It's an extremely stable protein. Or it may fall apart easily, right? You could do a detrimental mutation that prevents proper folding, or it's folded, but it's in a weaker bound state, or a weaker folded state, right? A higher energy folding state instead of being a lower energy folded state. It could cause it to fall apart easily. Right? What about the active site? Like I mentioned earlier, right? Active site can change the shape, size, charge, etc. It can modify the entire enzyme, right? You can go from doing one type of reaction to potentially doing a different type of reaction, or a much slower form or a much faster form of that reaction. It can also affect solubility. And that is often the case when you're in a research lab, right? When you make a mutation in a protein just to see what it does to the enzyme activity, you have no idea how it's going to affect your purification protocol, for instance, your thermal stability, right? Your protein folding, right? So often when you make a change in a protein and then you purify it, you often do a yield comparison, how much protein you got per original culture. You'll often see a decrease. And this could be either due of one to two things. We do a plethora of things, but mainly we look at two things, right? Folding and stability, or solubility, right? And these often, without doing further studies, can be mistaken for each other, right? You can imagine if you purify a protein, you end up getting maybe half the yield, right? And you could be thinking, oh, well, it's just not stable anymore. It's less stable. I changed an amino acid. But it could be just the fact that maybe it's more stable, or maybe it's fine. It's the same stability, same folding, but an argue of solubility is preventing it from being soluble. So you're just losing a lot of it during the insoluble portion of extracting the total protein from cells. So how do you determine which enzyme, wild type or mutant, is most effective at degrading the substrate RNA, right? Enzyme kinetics, right? The whole point of the talk, right? The culmination of a majority of the work that you've been doing, right? There's two steps to studying proteins. One is structure and folding and stability. The other one is enzyme kinetics, right? So what do we define enzyme kinetics as? Well, it's the study of the rate at which an enzyme converts substrate to product, and this can be under various conditions, right? We typically try to model it under physiological when we're studying a wild type enzyme. Right? But what we can get out of this is it explains how the enzyme works to a degree, right? Structure and functional studies tell us that. They tell us how an enzyme particularly binds a substrate. However, it doesn't tell us how fast it happens, right? Is there a rate limiting step somewhere? Is there a transition between when it's bound to doing it, the second half of the reaction? So this kind of really explains, we can get some key insight into how these enzymes work, right? It can also help us predict how enzymes behave. It helps us understand how mutations affect function, right? And so you'll often see when you get a new enzyme of interest or a new protein of interest, you're gonna first obtain it, you're gonna clone it into your organism of interest, or easy modeled organism, something like E. coli or yeast, or if you're studying plants, something like Arabidopsis or tobacco. And you're going to overproduce the enzyme or overexpress it. You're gonna obtain it via various purification techniques, right? And then once you have it, you're gonna do structure, you're going to characterize it, you're going to do structure, folding, stability, enzyme kinetics, then you're going to make mutations to see how it affects it, right? It's the ultimate, it's the penultimum of biochemistry is, I have this wild type enzyme, I'm going to change it, how does it affect it? All right, so enzymes are catalysts, right? We mentioned this earlier, right? They're in industry, they're known as bioreactors, right? They help these huge vats of substrate and coenzymes and metal ions that are required for the enzymes to operate. They just start to pump out, bind a substrate, transform it, pump it back out as a product, and just repeat that over and over again. So there's a couple things that are specific to catalysts or biocatalysts, right? Enzymes that are different from things like heavy metal catalysts. One is that something they share is that they increase the rate of the reaction by decreasing the amount of energy to reach the transition state. Now, this is something you have absolutely heard before, whether it be in something like Gen Chem or Orgo or even biochemistry if you're concurrently or have taken it, right? And you've noticed this from when you look at your reaction energy diagrams, right? Whether it's an exothermic or endothermic reaction, you have that huge hump that you have to clear at the apex of the graph. And that right there, at that apex of that curve, is the transition state, right? And they're called transition states because they are transient species. You will almost never isolate them in vitro or in vivo because they exist transiently. Once they're made into the transition state, they immediately fall into the next lower energy state, right? They act repeatedly to increase the rate of the reaction. It's very similar to organic catalysts, except that they are used up, right? That's the next part, not consumed by the reaction. That's the key advantage of being able to use an enzyme versus something like a heavy metal catalyst, because they are not used up. As long as you keep providing co-substrate and coenzymes, they can sit there and repeat the process. Unlike something like a heavy metal catalyst, you have to keep supplying that, and it becomes very, very expensive. Enzymes, unlike organic catalysts, are often very specific. Now, I say that in parentheses because they can either be very specific or they can be what we call promiscuous enzymes. RNase is a very specific enzyme in the sense that it only binds RNase, chews it up, spits it out, you get RNA fragments, right? Often enzymes only promote a singular particular reaction, right? One particular reaction. And even though some enzymes are promiscuous, they will often do the same biochemistry, right? The same chemistry is happening on the substrate. Maybe a different substrate may result in the same product or a different product, but the reaction itself is often the same. For instance, when you have things like reductases, there are often something during reductases that are very, very substrate promiscuous. They can bind to many substrates, but they perform the same biochemistry. They reduce double bonds in it. Insert hydrogens, right? Reactants are also called substrates. You've heard me talk about this before. We don't call them reactants anymore like you would in a traditional organic chemistry class or even Gen Chem or P-Chem. We call them substrates, right? These are substrates. Proteins have ligands when they bind them in a non-enzymatic way. Enzymes bind to substrates to make products. And I'll use that word substrates and products from here on out. Now, whenever we talk about enzyme kinetics, anytime you will often synonymously hear enzyme kinetics in place with Michaelis-Menten kinetics, or something like steady state kinetics or steady state Michaelis-Menten kinetics. There are all these different combinations. And it is the go-to way of measuring and modeling how fast an enzyme is producing product dependent upon the amount of substrate. So here I have a Michaelis-Menten plot. They will always generally look like this, where you have on your x-axis the substrate in millimolar, or any form of a concentration, whether it be in molar or mg per mil or mass per volume, right, on the x-axis. And then on the y-axis, you'll have initial velocity, right? This will either be represented as v and typically have units of the x-axis divided by some unit time, right? It can be minutes, seconds, hours, depending on how fast or how slow the reaction is. Now, when looking at the graph, there's a couple of key things. They will always look something like a hyperbolic curve, right? You'll come up, you'll have this high increase, this drastic change in slope until it eventually starts leveling off as you increase up the x-axis. You keep increasing substrate until eventually the curve will plateau at the top. Where this plateaus is what we call Vmax, or the highest rate of the reaction, or the highest initial velocity of the reaction, right? So maximum velocity of the enzyme. At half Vmax on these graphs, the x value corresponding to half Vmax is where you will find and call the Km. This is what's known as the Michaelis constant. It's often interchanged or synonymously used and as an interplay between Kd and Ka and Km. However, Ka and Kd are not identical to Km, but often you will hear these interchangeable, right? When we talk about enzymes with different substrates or different enzymes reacting with the same substrate, we'll often compare their Kms and say, oh, this one has a higher affinity for it. Right, and for all intents and purposes, we're going to do the same thing in this class, although if you are concurrently taking biochemistry, you might hear specifications on how to interpret KMs along with KDs and KAs from your respective professors, right? But here in this class, we're going to treat it as if it's very similar or proxy to KD and KA, right? So this KM is the substrate that gives half maximal reaction rate, and that's simply, you can always remember that by looking at a Michaelis-Menten graph. At half Vmax, your X value is your KM. Right. If an enzyme has a small Km, it achieves maximal catalytic efficiency at low substrate concentrations. And that's very important to understand because when you go to do enzyme design or rational enzyme design, right, that's the whole point. If you're trying to make more of a product with this enzyme, right, it's slow in nature and you want to make it faster, well, then you would need to do different mutations, different structural designs to achieve a lower Km, right, among many other optimizations you would do with Michaelis-Menten kinetics. And you've seen one of these graphs before, right? And essentially this is figure 5 in the Grainer, Stoffel, probably butchering those names, paper, right? And this is the Michaelis-Menten plot, right? On your x-axis, you have the substrate, RNA, right? It's concentration RNA, it's RNase. So the substrate is RNA, backs up, chews it up, right? And this, like I said, it can be in different units of concentration here. We have a microgram per milliliter. And on the y-axis, we have the initial velocity, right? V in those units of concentration of RNA, right? Microgram per milliliter per minute. Right, it's a unit of time. And look, you have the hyperbolic curve, you have your data points, it appears they're replicated in triplicate, right? And this is a Michaelis-Menten plot. However, tens, 20, 30 of years ago, when mathematical modeling or software was not readily available to model Michaelis-Menten, these hyperbolic curves, and doing so would take months, sometimes years to find the right one and plotting it and seeing if it would work, you would often use a Lineweaver Burke modeling, right? And what's called a double reciprocal plot, right? And all it is by taking the substrate, taking 1 divided by the substrate for all values and 1 divided by the initial velocity and plotting those on an XY axis as well, you end up getting a linear transformation of your Michaelis-Menten data, right? And the reason this is easier is because to the eye, it's very easy to glean some key components of Michaelis-Menten studies, right? So you'll always notice these lines, even though your data is always typically in the second quadrant of your coordination plane, right? Your second quadrant. You will always extend that line down and through the x-axis, and that's because located at the x-intercept, we have the negative reciprocal of km, right? -1 over km, right? That Michaelis-Menten constant. At the y-intercept, we have 1 over Vmax, and the slope of this line will always be equal to km over Vmax. Now, there's not really a trick in remembering that outside of just memorizing it. And it's very easy to memorize and then get all this information out if you just memorize the equation used to model that linear line. And it's not that hard, right? This is essentially y for 1 over V naught equals m, your slope, km over Vmax, multiplied by x 1 over s, your substrate, plus b, right? Y equals mx plus b. Always what we use to model linear lines, right? And this is all it is, right? It's just taking the Michaelis-Menten modeling equation and doing a double reciprocal of everything. And this is what you get, right? So the drawback is the points tend to be very clustered on the graph. And this introduces, when you go back and do this mathematically, will introduce quite a bit of error to your data. And it's why we don't really use this anymore, simply because, right, software has boomed in the past, 10, 15, heck, 20 years now, right? We have the readily available software programs and code that you can apply to a data set and get a very, very accurate model for is relatively easy now and far more accurate than it used to be. And that's why we typically just use Michaelis-Menten plots now instead of doing also Lineweaver Burke plots. But for this class, it's very easy to give you a Lineweaver Burke plot, give you one of these 3 values, and ask to calculate the others. Right? Make sure we can test you and you understand how to interpret Michaelis-Menten data, right? So we can't do enzyme kinetics in depth, right? Ribonuclease, why can't we do that? Ribonuclease SA is a very stable protein. It's great for classwork, right? For instance, when you purified it, you're able to keep it on the column for almost 2 weeks. Right? You cannot do that in a research hub. If like for me or any other TAs that you have had or had prior in this department, if we work on enzymes or proteins for that matter, we have to purify them that day that we open the cells, typically. Maybe within 2 days or a couple of days. We don't get, a week, 2 weeks time. We would lose all our protein. It would unfold, it would aggregate, it would, it would form inclusion bodies, it would precipitate out of solution eventually, or it might just stick to the column indefinitely and we just lose all of it, right? You have to recharge the column matrix, right? So great for you guys, not really great in a typical lab setting. Also makes it hard to do something like folding and stability studies because it's so stable. It's a great system for studying stability with protein folding, right? Because it's so stable, you can denature it and it'll quickly fold back. You can say that via CD or DSF or anything like that. However, it's a lousy model system for studying enzyme kinetics. And this makes it very hard to study enzyme kinetics, right? We can only calculate 2 kinetic parameters, Vmax and Km. There's no competition studies, right? We can't say, oh, well, what happens when we bind the substrate and increase the temperature, right? How does that affect the curve? Right? Does it affect the reaction rate? And it's not really going to because RNase SA is so dang stable. Right? By comparing these parameters, we'll be able to determine if changing a negative charge on the surface of RNase SA, right, Vmax and Km, has any effects on its kinetics. Right? And that's all we can really do with RNase A. Right? It makes it so very easy to study in a lab like this when we have short amount of time, or in this case, when we're all quarantined away from our house or from work, from school. Right? So we're going to use Km to compare wild type and mutant enzymes. So you have calculated the amount of protein in your samples, or we're going to do this for you and give you that data. And 2, the enzyme activity RNase interaction. Again, we would have done this in lab, but this data has been given to you now. So we define 1 unit of enzyme activity as the amount of RNase that leads to an absorbance change of 0.01 per minute, right? At 25 degrees Celsius, at a substrate concentration of 0.8 mg per RNA, or 0.8 mgs of RNA per milliliter. For the Lineweaver Burke, we're going to describe the units of the enzyme you used in this assay, not just 50 microliters of the enzyme, right? And it's an important part is to determine the effective charge on the enzyme activity, right? That's the whole point of this, right? You're going to get data to look at the structures and folding and stability of it when you change this, but how does changing the overall charge of RNAs affect its enzyme activity, right? The whole point. So step 3, we're going to calculate Vmax and Km. You're going to vary the substrate. Again, we'll be giving you data for this from 0 to 500 micrograms per milliliter of RNA. In a lab, is what you would do, right? You would sit down, you'd either use a spec or some instrument to measure a change in some observable signal in your reaction, right? So we vary the substrate concentration. We start always with none as a control, and then work our way through varying concentrations, in this case, 0 to 500 micrograms per mil of RNA. Right? And you're going to use 4 units of enzyme, right? If it's important for McKelly's Menten kinetic studies, no matter what, that you, prior to this, you always have to determine the optimal amount of enzyme to use in a specific time frame, right? That's probably the longest part of doing enzyme kinetics is finding this optimal enzyme concentration, right? In this case, we're going to use 4 units of enzyme, right? And you're going to do that. And it's important in McKelly's Menten kinetic studies that you never, ever alter the amount of enzyme you're using. You only change the amount of substrate you're using. Right, you'll notice in those plots, we never indicated a change in concentration of the enzyme, right? That's because when you do McKelly's kinetics, you're always supposed to report the amount of enzyme you used for your studies. So note that your RNA concentration curve showed as RNA concentration increases, sorry for my phone, absorbance increases. Turn off my phone real quick. Okay. So T equals 0 must increase as RNA increases in negative charge, or you have to redo it. Right? So you're going to graph the data as a Lineweaver Burke to determine what data points might be a problem. Normally you would show it to us before you leave. So during class time, right, we'll all have, well, it might not be me, it might be somebody else for this particular lecture, but we're going to be available during your class times via Zoom to help you answer any questions. You can screen share, you can send screenshots, we'll be able to help walk you through it and any questions you may have with this. Right? So you can show it to us during that time. We can give you some feedback on it and tell you if you're looking good or if you try to point you in the right direction. Right? You're going to include your Lineweaver Burke graph in your final paper. Right? This is very, very important. If you're in the writing section, you must include this Lineweaver Burke graph in your final paper and your big table from worksheets 8 and 9. Okay. So for finally, you're going to compare the mutant and the wild type by sharing data, right? So if you remember in class, we had those 2 bench tops and that were filled, first 2 from the farthest door nearest the elevators were wild type, and then the other 2 tables closest to the bathrooms in the major part of bio were the mutant, right? So you're going to compare Vmax, and if everyone uses the same amount of enzyme, 4 units, right? It's very, very important. If somebody doesn't use 4 units, right, that's the nice part about doing this now online is that we're going to give you that data, so it will be correct. Right, you're going to use the Km independent of enzyme concentration. If the enzyme is more active, you're going to have a lower Km. If it's less active, you're going to have a higher Km. As everybody, you need to understand how and why that is, right? Think about what Km means, right? Km is the amount of substrate that gives you the half maximal velocity. Right? So if you have a more active enzyme, you should need less substrate in order to achieve that maximum velocity. Right? If it's less active, you will need more substrate to reach that same half maximal velocity. You're going to do this when you turn in both worksheets. Online, this might change, so please be on the lookout from emails from Dr. Ayers, and please keep checking eCampus to understand how we are going to have you do this, when they will be due, and where you're going to turn them into. For your calculations, you're going to use 1 over V initial velocity for the Lineweaver Burke plot. Your x-axis will be RNA concentration to the -1 or 1 over. Please, I would prefer you to put RNA concentration to the -1, right? All this means is the reciprocal of RNA concentration. It's very, very important. You're going to use the concentration, and your units will be in microgram per milliliter to compare the data. Just the same as the paper we assigned you already, right? Your y-axis will be initial velocity to the -1, okay? And don't just put initial, you can put initial velocity, you also need to include your units, right? And your initial velocity is going to equal the change in absorbance, right, which is a unitless number, per minute. So it's going to be units of A688 minute per minute. That's going to be the -1. You must have enough enzyme to repeat it as needed, right? So this more or less isn't going to really affect you guys because you'll be given the online data. But if you were going to do this in class, you do not have to do osmotic shock acid precipitation. Impure samples alter kinetics, right? You must have enough good points. You need to your setup is identical to previous assays, right? Your RNA is 0 to 500 micrograms per milliliters, more at a low concentration. Your enzyme will always be 4 units. MOPS, which is our buffer system, at varying concentrations, per varying concentration, and methylene blue for 500 microliters, right? That's what the indicator will typically be measured by the spectrophotometer. Your final volume will be 1 milliliter. At 10, you would incubate the reaction for 10 minutes before adding the enzyme, and you're going to mix all solutions before using, right? You never come to a lab bench. Even so, what I do when I come in to do enzyme kinetic studies or any type of kinetic study or enzyme reaction, I will typically prep unless otherwise absolutely needed to be prepped right before a reaction. I'll usually prep everything I can the day before. And when I come in the next day to start my reactions, because they sometimes take many hours to complete, I will mix everything very well and thoroughly before using it that day, because things do settle, right? You can get gradients if you have multiple things mixed into your reaction. You're going to convert your data into a Lineweaver Burke plot for your paper, like I mentioned earlier. Right? You're going to parameters to calculate on worksheet 9. You need to calculate specific activity. And this is defined as the amount of activity per unit of protein, or in this case, enzyme. So it has units of activity per milligram of protein. There should be a higher number when pure. So if you remove contaminating proteins, that increase the denominator, right? So if you have impure protein sample, right, you have multiple proteins, you're going to have an overall higher amount of protein in solution, and that way your specific activity would then be lower, right? You need to calculate. Lastly, none of these are irrelevant, but I just want to remind you guys, please make sure to wash your hands if you ever keep going out, or just wash them frequently as instructed. You know, masks don't really work because the virus is much smaller than your average guarding mask that you pick up from the dollar store, or if you paid $300 for it on Amazon. And lastly, please keep up with your studies. I know this is a very awkward time. Some people do better in class, so this might be a little bit jarring, but like I said, I and the rest of the TAs and Dr. Ayers are here to help you guys try to succeed as best as you can, but you got to put in that effort yourselves as well. So make sure you're staying on top of your studies, and please stay healthy, everyone. Thank you, and I will see you at Zoom. And remember, my office hours for anybody, whether you're in my section or another one, will be Wednesdays at 9:30 a.m. I will be on Zoom. I will send out via email that code, or we'll put it on eCampus. I'm not sure yet, and you guys can come join, and I will be available from 9:30 to 10:30 to help you. I'm still available by appointment now. I'm working from home. So if you want to set up a time to just meet with me one-on-one via Zoom, I can walk you through some things, some concepts if you're having a hard time understanding them. And like I said, I and myself, the other TAs and doctors, are here for you to make sure that this goes as smoothly as it possibly can, trying to teach a lab online. So with that, I wish you guys good luck, and I will see you soon.